All right. Well, I'm the odd chicken out, so to speak, here with all of the ruminant and dairy interests. But I really want to thank the organizing committee for inviting me, and 2 years ago, I guess. And when I look out right now, I think I see everybody as survivors. And by surviving, it means you're either smart or lucky. So with that, okay, so I'm going to give, this is the outline of the talk. So an introduction, I'm going to talk about feed efficiency in pedigree male broilers. This is a elite line of broilers that was developed by one of the commercial genetic companies. It's about 30 miles away from University of Arkansas. Through these studies, we got into looking at mitochondria hormone receptors, and it led us to progesterone signaling, which I'll talk about. So getting ready for this talk, I wanted to go see what was a little more out there rather than just mitochondria. And so looked for papers, a couple of papers that had evidence of other hormones in skeletal muscle. And so there's some in the literature. And then we also did some global expression studies, and we did shotgun proteomics, microarray, and RNA-seq. And what I'll say is you can drive yourself crazy with that. But all of those studies were done on the same set of tissues. So from the same experiment. And so we didn't have a variation from study to study. We used ingenuity pathway analysis. And if anybody's familiar with this, that will drive you crazy. So it's amazing that I'm even standing up here. And then what I want to do is kind of finish with some summary and thoughts. So we're all in agriculture. We know that feed costs a bunch when you're raising an animal to market weight. So in the broiler industry, it's about 60 to 70% of the total cost of raising an animal to market weight. We, for some time, have been studying the role of mitochondria in the phenotypic expression of feed efficiency. And so the animal model was this. The genetics company raised, about 3 times a week, they would raise 300 males in a pen under identical situations. And then from that, they took the top 100 out of that 200. And then what we did was we took the top, so that the phenotyping for feed efficiency occurred after that and in the top 100. So they put them in individual cages and phenotype for feed efficiency from 6 to 7 weeks. And then out of that 100, which is the top third, we took the top 8 and bottom 8 for our studies. And so this is the first study that we did. And what this is showing is the birds ate the same amount of feed during that week. So there's no difference in feed intake, but they gained more weight with the high feed efficiency. So we had a big difference in feed efficiency. And the starting weight, I should have put that up here, but the starting weights were similar also. So it wasn't like there was a different starting weight. And Gordon Karstens at Texas A&M asked me to speak for a meeting. And so we got a paper out of this, and people may be a little more familiar with residual feed intake. So the red triangles here, those were the low feed efficient phenotype. And this is like the 100 birds, all of the dots in there, triangles. So the red ones were the low feed efficient, and this would also be the high RFI. And the green ones were the high feed efficient or low RFI. So this was the first study we did. And it was really kind of funny because the company brought us the birds over, we acclimated them to their cages, and they were really pretty similar to what they'd been in. We gave them about 3 or 4 days of acclimation, and then we started doing functional studies. And I said, don't tell me which bird is which, from which group. I said, just paint one group blue and the other group just leave alone. And so we did 3 birds. We had 2 of the blue group and 1 of the white group. And I called up the geneticist and I said, I know which group is which. And he said, there's no way. And it turned out the functional difference was huge. So this is the RCR's respiratory control ratio. ACR is something that's very similar to that. And then there's oxidative phosphorylation, ADP to oxygen ratio. But the coupling was really different. The RCR was very different. And this is showing it in mitochondria from breast muscle and leg muscle. And then we did a whole bunch of studies, and we did them in different tissues: breast, leg, liver, duodenum, and heart. And sort of as a summary, what we found consistently was in the high phi deficient, we had better coupling, we had lower electron leak and reactive oxygen species production. And we also had lower protein oxidation. So the high phi deficient birds had less protein oxidation than the low phi deficient. And so we jumped into the deep end of the pool and started doing transcriptomics and proteomics. And so in this particular paper, I began collaborating with Nick Hudson and Tony Reverter from Australia. And we were looking at the data, and we started seeing it, and it was like there's more proteins or more transcripts that were higher numerically in the high phi deficient compared to the low phi deficient in the mitoproteome. So the first one was the shotgun proteomics, and so there was a very significant binomial P difference between the 2 groups. And then we also did it with an RNA-seq. And so this is the data. What's showing up there is just the mitochondria transcriptomics. And so the blue were the high phi deficient numerically higher, because we didn't, and I'll explain why, we didn't do a fold cutoff, or we just looked at, it was yes, no, was it higher or not? And so this led to, this was part of this paper on progesterone signaling. And this is with Hudson and Reverter. I got a small grant to go down, fellowship, to go down and work with those 2. And we developed a grant, which ended up getting funded by USDA. This is Nick with his son at the time. From a Zoom. We were doing Zoom before Zoom was cool. And so that was his son. His son's now 8 or 9. And this is Tony Reverter. He's a statistician, mathematician. I believe he's also a reproductive physiologist. So they had developed this thing that's called regulatory impact factor. And in this, again, there was no difference. We don't do a cutoff. So you take all the data, and what they're trying to do is to see the interconnectedness of genes or proteins. So you could have something that was not significantly different, but it's having a big major effect. So it's kind of like operating under the radar. And so this method is described in these 2 papers. And so we were using that to look at the data. And this is the top regulatory impact factors. And out of the 10 that are shown here, 4 of them said that it was related to progesterone signaling. And this is how they would calculate both an RIF1 and RIF2 and plot it out. And so the most significant ones are the ones labeled. And so we're going, what the riff? You have progesterone signaling in breast muscle in immature pedigree males. I thought there was something really screwy here. So how could this be playing a role? And so I started looking at the literature, and one of the first things I found, this group had done a study of pediatric brain injury. And so they had animals that had the brain injury, and then another group that got progesterone before the injury. And so progesterone improved the mitochondrial function, and the differences looked remarkably similar to the data that I showed you. Then the next thing I found was that there were a number of hormones that are interacting with the mitochondria. So there's mitochondrial hormone receptors. And so now we've got a couple of things to look at. And so the classic, and I'm not an endocrinologist either. I'm a physiologist, so don't quiz me on all of this. But so the classical way is the hormone interacts with the receptor that gets carried to the nucleus, and you have the hormonal response element. And so now that's in the nucleus, but you also have it in the, I need a drink of water. It's been a couple of years before speaking in front of a group, and I wanted to wear this so that if you see my wife, you can tell her that I did wear it and you did see me. But if this was on a Zoom, yeah, if this was on a Zoom, there's no telling what I'd be wearing down here. So everybody's been there. All right, so thank you. So there's mitochondrial hormone receptors. So these hormones can directly interact with the mitochondria without going through the nucleus, which I thought is really pretty cool. So we wanted to see if this were the case in avian. So the first study here was done with QM7 cells, which Dr. Hargis back there would call not real because it's an immortal line. So anyway, so we did staining of these cells, and we found the progesterone receptor, but then we also we identified glucocorticoid, thyroid, insulin, and estrogen receptors on these QM7 cells. So the blue is the stain for the nucleus. You have the receptor being labeled. Mitotracker is specific for the mitochondria. And so when you merge them, if you see yellow to orange, that means that the receptor is on the mitochondria. And we saw that for each of these. So then we wanted to see if it were real. And so we had breast muscle tissue, and we did differential centrifugation. We identified the nuclear fraction to make sure that we were not getting any bleed over from the mitochondria. So mitochondria, there was nothing labeled there. The cytoplasm, and then voltage dependent activated channel is a protein that's specific to the mitochondria. And so we were able to show that we were able to get very good separation in both broiler and quail muscle. And then we went ahead and did the Western. When I say we, Dr. Lassiter did all the grunt work on this. He's the brains of the outfit. And so we did progesterone receptor, glucocorticoid receptor, thyroid, insulin, estrogen, and we identified it, that it was present in breast muscle tissue and in broilers and in the quail breast muscle. So we got to that point. So we have identified that there are these present and they're in muscle. So now I wanted to take a moment and kind of back up a little bit. So what about hormonal signatures in muscle from literature? And so this is a paper, and what I'm doing is highlighting the thing that they looked at. So this is oxytocin. So Dieger et al., which had Hudson and Reverter on the paper, They found that when they did a implant, one of I think it's trobrinolone something. They gave that implant. There was a 27 to 90-fold increase following estrogen and progesterone implant, and then there was a 50-fold elevation in oxytocin coming out of the muscle. So the muscle is producing oxytocin. This is another one looking at oxytocin precursor mRNA. Dr. Lassiter, he did another study with Erection, which is a hormone in the hypothalamus involved in regulating feed intake. And then there's, these are a couple of review papers, and they're showing oxytocin being involved. But in this last one, oxytocin, osteocalcin, interleukin 6, and then nerve growth factor, bone, brain-derived neurotropic factor. So there's several things that appear to be coming out of the muscle itself. And then in another review paper there at the end. So Kentu did this study looking at Erection, and this was in collaboration with Sammy Dridi in our department. So the QM7 cells are secreting Erection. And then when we did some studies with seahorse to look at mitochondrial function, so Erection decreased uncoupling protein. And uncoupling protein is one that uncouples the mitochondria. So you get the transport chain running, but you're not producing energy. And then looking at it with the seahorse and dissecting out the different components of mitochondrial function, proton leak was diminished. So we've got a decrease in UCP and decreased proton leak, which makes physiological sense. So the next thing we're doing, so we've got all these big data sets, and so what I wanted to do here was exploit another part of the Ingenuity pathway analysis program. There's all kinds of stuff that this program does, and like I said, if you want to turn into a goldfish, just do this and you'll have lots of goldfish moments. The shiny lures going by. So upstream regulator analysis, it's based on the prior knowledge of expected effects between transcription factors and target genes. So IPA's got this like over 6 million literature citations that they've put into this program. And they look to see, you know, if there were 2 hormones or 2 transcription factors, what was the relationship between those 2? So it examines the known targets. So if you have a upstream regulator, it can be affecting downstream events. So it's going to be looking in your data set relationships between the upstream and downstream target molecules. And so it compares this with the data that's been curated in IPA. And then it assigns, it's called an activation Z-score. And so this activation Z-score, and it also gives a p-value, which is p-value of overlap, or Fisher's exact p-value. And then based on all of this, it makes a prediction. Is that transcription factor being, is it predicted to be activated, or is it predicted to be inhibited? And so this is the legend. So red, when you see this, it's actually a measured or a gene or a protein. And so it's upregulated, and in our case, upregulated in the high feed efficient phenotype. And then the green is downregulated in the high feed efficient phenotype. So it's comparing the 2 groups, or the other way to look at that is if it's downregulated compared to the high feed efficiency, it would be upregulated in the low feed efficiency. This, the orange and blue, these are designated predicted activation or predicted inhibition. And then it generates the activation Z-score, and as well as a p-value for that relationship. And so this is from the shotgun proteomics study that we did. And I pulled out 4 of these that showing progesterone, insulin-like growth factor 1, insulin receptor, and triiodothyronine. The 1.94, 1.91, this would be like a p equals 0.06 if you're doing a t-test. So those 4 were all activated in the high feed efficient phenotype. And just, this is part of a figure that I had, and it was just showing that what effect that these might be having on the mitochondria. Whoops. It also, we had indication that mitochondrial biogenesis was likely going on because we had an elevation in AMPK that would be stimulating PGC1 alpha, which is involved in mitochondrial biogenesis. And so this is an upstream regulator network. So progesterone is in the middle, and around it are downstream target genes. And this is what IPA was doing a couple of years ago. And I put the IDE is insulin degrading enzyme, and there's an isoform of this that actually helps getting proteins into the mitochondria. So this analysis supports what we saw in the regulatory impact factor analysis. And now what they've done, because they have too much time on their hands, I think, but they've now been able to link upstream regulators with downstream functions and effects. So this was a new addition to IPA about a year, maybe 2 years ago. So here's the upstream regulator, target molecules, and then the downstream functions from this single network. And so all of the orange are going to be predicted to be activated in the high feed efficiency, or this would be inhibited in the low feed efficiency. And I will point this out, is that 4 out of 6 of these, and I didn't see this right off the bat, but 4 out of 6 of these are targeting cholesterol or aldosterone pathways. And here's another one. This is with IGF-1R and insulin receptor. So it's predicted to be activated. Now in this case, the blue, these are all predicted to be inhibited in the high feed efficient phenotype. And so apoptosis, hypotrophy, not hypertrophy. It would be inhibiting mitochondrial dysfunction, and it's inhibiting mitochondrial uncoupling. All of those would favor a better mitochondrial function. And so not to leave it alone, I decided, okay, because all of these things are going to be working in concert with one another. So what would this mean if you put these all together and started pulling in these downstream effects? And so these are ones that were inhibited in high feed efficient. It's disorder of lipid metabolism, inhibition of apoptosis, vascular cell apoptosis, and mitochondrial dysfunction and hyperlipidemia. All of those were predicted to be inhibited. These are all basically a hypothesis. Every one of these, it's all predictions. So I'm not going to go out on thin ice at this point yet to say that this is the be all and end all, but it is pointing towards certain mechanisms that I think would be pretty important to look at. And then these are the ones that are activated in a high feed efficient. Metabolism of protein, vasculogenesis, hormone synthesis. You can look at the rest of them. And the one down here, it said actin skeleton disruption. We'd actually written a paper about 6 or 7 years ago and hypothesized this, but it was on for a different reason. So this was in, we'd hypothesized it in 2012, 9 years ago. Time flies. So now what I did, getting ready for this, is I went into our data and pulled up all of the hormones that I could see that were in there. And so there's aldosterone. We got a prediction, mild sort of prediction of inhibition in the RNA-seq study, but we had a very strong one in microarray, was minus 2.09. Angiotensinogen, the only one that in this particular data set was a brain-derived neurotropic factor. And so that's a neuro-related hormone, and it's showing up, or transcription factor, and it's showing up in the muscle. Nerve growth factor, prolactin, parathyroid hormone. And in the first study that we did with the microarray, corticotropin releasing hormone was downregulated in the high feed efficient phenotype. So we're seeing all these little tantalizing things that are suggesting there's a lot of hormone stuff in there that you wouldn't expect. How am I doing? This is the brain-derived neurotropic factor. There was a real mild predicted activation in the proteomics, but they were both predicted to be activated in the same direction. This is looking at it in terms of downstream effects, and it's not real strong, but it's interesting. Growth of axons, so it's picking up a lot of stuff related to nerves, but the lighter color on this means that it's not quite as strong as the real dark colors. And then looking at the literature again, I'm going to highlight this, the black little bars there. This is aldosterone metabolism. And so the title of this paper is Myeloid Cells Are Capable of Synthesizing Aldosterone and Exacerbates Muscular Dystrophy. And so all of those different steps in aldosterone synthesis are there in the muscle. And so the whole tissue expresses all of these genes. And then I went in and looked at the data again. We found 6 out of 6 that were there were upregulated in the high feed efficient that would go from in cholesterol synthesis. So here's a summary. Right now, progesterone signaling is involved in phenotypic expression of feed efficiency. Mitochondrial hormones receptors, we identified them in cells and in tissue. And we got the receptors, sorry, I jumped ahead. Mitochondrial receptor proteins were identified for progesterone, estrogen, corticosterone, thyroid hormone, and insulin in breast muscle of broilers. We had some upstream analysis talking about from the proteomics data showing insulin receptor, insulin-like growth factor, progesterone, and T3 were predicted to be activated in the high feed efficient. And then upstream analysis of transcriptomics data showing potentially a whole bunch of other ones. And I think that these hormones might be operating under the radar, that this could be going on potentially within the tissue itself, that you'd never see it outside in the circulation. Although in the case of oxytocin, it was very high. So, and this last one, predictions represent hypotheses to be tested. And I want to leave you with this. I didn't like the way that this talk was ending up, and I went into like a bunch of papers that I'd saved and hadn't gotten ready to read, and this was the first one that I pulled up. So I rediscovered this paper. I had this since 2018, and I rediscovered it a couple days ago. So energy is required for life. Energy largely derived from mitochondria. Energy is needed to respond to stress. Glucocorticoids and steroid hormones are produced and metabolized by mitochondria. Mitochondria respond to neuroendocrine and metabolic stress mediators. And experimentally manipulating mitochondrial function can alter physiological and behavioral responses to psychological stress. So the neural circuits regulating social behavior as well as psychological processes are influenced by mitochondrial bioenergetics. And so this is kind of interesting. There's three figures that they had in here. So mitochondria do exactly the opposite of what chloroplasts do. Chloroplasts are making food and they're putting out oxygen. Mitochondria are sucking up oxygen and sucking up substrates. Stressor, you can have auditory, visual, metabolic, whatever pathogen is going to be interacting with information from the animal, which would be circulating hormones. It could be DNA, epigenome, proteins, et cetera, and memories. This is a neuro paper, so it's going to be totally focused on the brain. And so all of these are going to be involved in stress adaptation. So several of the talks had, and I don't know all these steps, and I'm not going to pretend that I do. This is looking at the adrenal gland. And so what is going on is steroidogenesis is occurring in conjunction with the mitochondria. So there's this star protein, which stands for steroidogenic acute regulatory protein. And then it's going through a pathway of P450 for a step or two. Then you get the pregnenolone. And then it actually will migrate out into the endoplasmic reticulum for a couple steps. And then it comes back into the mitochondria. And in this case, in the adrenal gland, cortisol is being produced and is being secreted by the adrenal gland out into the plasma. And then there's also an interrelationship with the catecholamine pathway. So you've got two important stress hormones that are being linked in one way or another to the mitochondria. It says catecholamine pathway occurs mostly in other cell types, but it has the tyrosine hydroxylase and monoamine oxidase A and B are anchored to the mitochondria. And so here's the last thing. What am I supposed to be done? Three? Oh, all right. I didn't know how long this was ever going to take. So in this is the final figure. So in response to stress, cortisol is being produced by the adrenal gland, and they call this the mitocentric model. So it's the mitochondria is the be all and end all of energy substrate mobilization. So it's saying that there's stress, animals got to do something about it. And so you end up with increased lipolysis, gluconeogenesis. You have increased energy released from organs like fat or the liver. And in this case, it's increasing glucose and lipids. And these will circulate back to the adrenal gland, which then is using it to produce more cortisol. But I think that there is something going on for muscle and other tissues where it can have just within the tissue itself, you could have this communication, tiny little bit of a hormone or being produced that would be interacting with mitochondria could have a dramatic effects on mitochondrial function. And so these are some of the thank yous. Dr. Lassiter, he got a master's and a PhD with me, and I kept him around for a postdoc. Dr. Kong, Dr. Dreedy, and Nick Hudson and Tony Reverter in Australia. This was supported, a lot of this was supported by USDA. And then I want to, a funny thing happened on the way to the mitochondrial forum. This is kind of a shout out to my mother and father who liked this play called Funny Thing Happened on the Way to the Forum. And so about 2018, I've been diverted, my energies have been diverted a little bit by, we were lucky enough to get a USDA SAS grant funded. And so I'm the project manager, project director of this. And this was a funny thing because on the same day, I had a colleague, Dr. Dreedy, walked into my office and said, there's this program at USDA, we ought to apply for it. I said, great, I'll help you write it. And he says, no, I want you to write it. And then I got a call from Shinjin Lee at Cornell, and he said, yeah, there's this program, we ought to go for it. And I said, great, you write it, and I'll help you, package it and do all that. He said, no, you be the project director. So we got funded, we're 2 years into it. There's some really cool stuff that's coming out of this. I'm really, really, really excited about. And one thing, I still got a minute or two. Go look up Turntide Technologies. It's a company that is based out of California, and they're producing motors through smart technology that are having a tremendous reduction in energy consumption. In other words, they're generating the energy a lot more efficiently. And the funny thing is that they, since this is a dairy audience, I was looking at a video that was in there, and they showed dairy barns and ventilation systems. They had partnered with a company called Best Artex, and so they're doing ventilation that is very much, helping dairy in their operation. I went up and visited their company in Wisconsin, and the vet that was there on staff was talking about how he wants to get air down for the calves. He wants to get the air down right at the floor so to dry out the bedding and reduce respiratory disease. And I went, that's chickens. I mean, that's exactly what we got to do in chickens. So we're going to try to partner with them, see if we can't do some testing of their systems, because this could really benefit growers and help lower carbon dioxide use production. Okay, now I'm done.